In 1901, a 51-year-old woman told her doctor, "I have lost myself." In 2022, investigators found evidence that one of the most cited papers explaining her disease may have been faked. That disease is Alzheimer's. The space between that first clinical encounter and this modern scandal spans 7 decades of medical neglect and a multi-billion-dollar effort to find a cure. This video explores how the bedrock biological truth of a disease can survive decades of human error, structural bias, and manipulated science. To find that biological truth, we start in Frankfurt, where Auguste Deter began showing severe memory loss, disorientation, and paranoia at a strikingly young age. Over 5 years, her condition deteriorated. When her doctor asked her simple questions, she would sometimes only reply, "I have lost myself." That doctor was Alois Alzheimer. When Auguste died in 1906, he examined her brain tissue and identified 2 abnormalities that still define the disease today. This microscope slide shows exactly what he saw. Those dark, dense clumps are amyloid beta plaques building up outside the brain cells, and the twisted filaments around them are neurofibrillary tangles of tau protein. But the history of science is rarely clean. In 1907, a Prague-based researcher named Oskar Fischer independently discovered the exact same plaques in 12 elderly patients, yet history would assign the credit almost entirely to Alzheimer. Emil Kraepelin cemented the name in his 1910 psychiatry textbook. In the eyes of the medical establishment, Fischer's contributions simply faded away. The biology was sound, but the recognition was social, a reminder that while proteins follow laws of nature, medical history is written by people. Inside your brain, the dominant amyloid cascade hypothesis begins with amyloid beta fragments drifting between neurons, slowly clumping into solid plaques. These plaques trigger a chain reaction. Inside neighboring neurons, tau proteins, which normally keep internal transport tracks straight, twist into chaotic tangles, disrupting transport and killing the cell. We know this sequence is accurate because of rigorous genetic evidence. Rare mutations in the amyloid precursor protein gene guarantee early-onset Alzheimer's. Furthermore, people with Down syndrome carry an extra chromosome copy containing that exact same gene. As a result, they develop Alzheimer's pathology at strikingly high rates and unusually young ages. As neurons die in the hippocampus, a region critical for memory, patients lose short-term recall. Over time, that localized damage cascades into language impairment and executive dysfunction. This sagittal MRI scan shows the structural reality of that atrophy in the medial temporal lobe. But today, a standard blood draw can detect p-tau217, a biomarker that identifies the disease's biological signature years before significant symptoms appear. This physical, genetic cascade is a measurable fact. It is a biological signal occurring entirely independent of whatever labels the medical establishment chooses to use. Which brings us back to a significant failure in medical history. Because Auguste Deter was only 51 years old at the time, her condition was classified by doctors as a rare pre-senile dementia. By separating early onset cases from elderly ones, the medical field created a consequential blind spot. For 70 years, millions of older people with the same brain pathology were dismissed as senile, an unfortunate feature of growing old rather than a specific disease warranting research. The correction finally came in 1976, when neurologist Robert Katzman published a landmark editorial arguing that presenile and senile dementia were the exact same killer. Katzman's reframing identified a neglected curiosity as an urgent public health crisis. The funding followed immediately, as shown by this climb from $5 million in 1980 to over $600 million by 2005. Changing the terminology didn't alter a single brain protein, but it completely redirected the world's focus. It showed how much scientific progress depends on how we choose to categorize a problem. That surge in funding created the conditions for a crisis of another kind. By the early 2000s, the field was suffering a lack of confidence after several failed drug trials. Into that vacuum stepped Sylvain Lesnay, whose 2006 paper in Nature claimed a specific amyloid beta assembly directly caused memory loss in rats. The findings had an outsized impact, receiving over 2,000 citations and helping steer a dominant portion of federal research budgets for 16 years. The data unraveled in 2022. Whistleblower Matthew Schrag and Science magazine investigator Charles Piller exposed apparent image manipulation across roughly 20 of Lesné's papers. By 2024, the co-authors of the original Nature paper agreed to its retraction. Lesné left his university that same year, though he did not agree to the retraction himself. This was a systemic oversight, where the scientific establishment built upon a mirage because it desperately needed a targetable culprit. But here is the vital nuance. The amyloid cascade hypothesis did not collapse when Lesné's paper was retracted. The foundational evidence, the APP mutations and the biology seen in Down syndrome, exists entirely independent of that 2006 paper, separate from Lesné's contested findings. One bad actor misled the field for 16 years. The eventual retraction, though delayed, demonstrates that independent scrutiny can eventually strip away a mirage, even after it has taken root. That scrutiny has led to real, albeit modest, progress. While older drugs only treat symptoms, new monoclonal antibodies like lecanemab and donanemab actually clear amyloid from the brain and slow the rate of cognitive decline. These new drugs carry real safety concerns, including brain swelling and microhemorrhages, requiring regular MRI monitoring and careful patient selection. From a brain examined by hand in 1906 to a blood test today, the science held up. Not every paper did. January Science tells the story behind the science, one disease at a time. Subscribe for the next one.